whenever you want to start, I'll share the screen. And we can start. Dr. Lea, you let me know when you want to start. Should we start?
Yeah, you can start, please, Dr. Kwa. Please.
Okay. Perfect. So we got for the last lecture of this series, and I want to thank again Professor Liao for organizing this, and all of you for assisting to this. I know that infection for many of you cannot be the most exciting subject, but I think in view of what is going on in the world, this lecture is more relevant than ever because we understand very little when pregnant women are in risk and to what type of infections they are in risk. So I think the subject is a subject for continued discussion, and I think we just starting to learn and understand what are the associations with and the risk for pregnant women. So infections represent, as we know, an important and frequent mechanism of disease in pregnancy complications, such as preterm birth, miscarriages, and fetal death. I mean, that's certainly well established. Now, we know that we have two types of really effects. Let me move here a little so you can see me. One of the aspects is associated with the effect of the infection in the mother. So during the pregnancy, if there is infections, especially viral infections, women are in risk even of dying. That's called the maternal mortality. And the second side is also the effect of the infection into the development of the fetus, as well as the well-being of the fetus. We know very well now that everything that affects the mother during the pregnancy is going to have an effect in the baby. The baby is not isolated from what is happening in the body of the mother. So again, I think this is an important concept. Everything that affects the mother affects the fetus. So let's start looking in general, what are the effects of the infection during pregnancy, the maternal effect. For many years, including this time of the pandemic with SARS-CoV-2, the COVID-19, pregnant women are more likely to experience severe complications, mortality associated with viral infections compared with the general population. And there is plenty of literature supporting this situation. One is, for example, you see I'm getting a message, maybe it's the slide is not in full screen. Okay, nobody told me, let me see if I can change it. Now it's in full screen. And that's better, okay. During the 1957 and 58 influenza pandemic, 50% of the deaths occurred to pregnant women. During the H1N1 pandemic, there was a high mortality occurring in pregnant women. And during the COVID-19 pandemic, the data, at least from China at the beginning, was that pregnant women are not in high risk. However, here in the United States, the picture has been completely different. And there is a high incidence of mortality in some pregnant women. There was a recent paper describing the experience in Philadelphia about a COVID-19 infected woman with a high number of mortality or complications in pregnant women. So it's, especially with COVID-19, has been difficult to define whether they are at high risk or not. The experience in different countries and in different situations has been different, which is also a challenge to understand what those differences are and what they are associated with. So an interesting aspect, and again, in the past, we don't know about COVID-19, the deaths of pregnant women were associated with bacterial complications. In terms of the fetal side, viral infections during pregnancy correlates with increasing fetal development of abnormalities. So infections like influenza has been associated with increase in the incidence of schizophrenia, autism, and more recently, of course, with the appearance of Zika viral infection, the connection between viral infection and fetal development associated with microcephaly is, without any doubt, a clear effect. So viruses can affect the development of the fetus. So the question still is how viral infections during pregnancy can affect the fetus and the mother. Because again, you have different circumstances. Some infections will have a terrible and detrimental effect, such as influenza. And then you have COVID-19 pandemic, where you don't see, again, at least in some places, that effect. So what makes, then you have Zika with microcephaly. So the question is, what makes, in some cases, the pregnant woman very susceptible to viral infections, while in other cases, they are even, they respond better than the general population. So we need to remember what I always present as a slide, that viral infection always induces an immune response. That immune response induces an inflammatory process, which is required for controlling the viral infection. Now, that's when the things start to differentiate. Is that inflammation a controlled inflammation in case of the pregnancy will lead to an eventual term labor, or that inflammation is an uncontrolled inflammation that then it will lead to fetal death and termination of the pregnancy. So again, what we are learning is that sometimes it's not so much the trigger, it's not so much the virus, it's the type of immunological response that we have against the virus. And everything goes through the inflammation. If it's a control, nothing happened. If it's an uncontrolled inflammation, we will have a damage and a termination and mortality, potentially in both, in the fetus as well as in the mother. So again, we have been studying multiple types of viruses in the lab for understanding the type of inflammations. We have used DNA viruses, RNA viruses, and of course, now we're trying to do different models for coronaviruses, which I will not talk today, although the data again is fascinating to the side. Okay, so just to recapitulate what we learned many years ago, for those who are not familiar with our work, we infected mice with MHV68, that is a herpes virus, is a DNA virus. We injected in the early pregnancy in the mouse. The mouse was 19 days, so 8.5 is the time of placentation, when the placenta starts to be formed. And we didn't find any effect. Yeah. So that injection of that specific virus in this mouse strain, the C57.6, did not induce a massive inflammation. There was no effect on the development of the fetus. As we say, there is no changes in the weight. So clearly, no effect of MHV68 on the pregnancy outcome. The placenta is infected. So this is another interesting aspect. When we look at the seed, this is PCR for the virus. This is the controls. And here you have the placenta, the deciduals and the placentas that were infected. You clearly see the presence of the virus, viral DNA in those placentas and those deciduals. So viruses do reach the placenta. And this is something that I want you to take, the message that I want you to take now. So you remember, the placenta is an organ that is infected by the majority of the viruses. The difference, so what is the difference? If it's infected with the majority of the viruses, what makes the difference with other organs? And here I think is the response to that. If we compare the viral replication or the viral titers in the stroma cells versus trophoblast cells, you see that they both are infected, as indicated before, but the stroma cells, the infection continues exponentially, and the virus at the end will kill the cells. And that is the reason you see this decrease. This decrease is because the number of cells have decreasing, they have been killed by the virus. On the other hand, and this is the fascinating aspect, the placenta gets infected, but then it's capable of control the virus. Yeah. So the replication of the virus is nicely limited, yeah, and maintain control, so there is no cell death. We look also in the tissues, multiple tissues of the mice, spleen, which is a classical target of this DNA virus, and indeed is positive, lymph nodes, placental decidual that I showed you before. And the surprise, the nice surprise is that the fetus is never infected. So the virus never reached the fetus. Infects all the organs around, the decidual and the placenta, but it will not go to the fetus. So what we see is that indeed there is an infection surrounding the fetus, the placenta, the decidua, but that infection is not capable to reach the fetus. However, remember what I told you, that everything that happens in the mother in some way will reach the fetus. So we are seeing in the placenta an immunological response to the presence of the virus. So the question was, is that immunological response, that inflammatory response, having an effect in the fetus, is reaching the fetus? So what we did with those, with mice, something that you can do only in mice, but not in humans, of course, is to take those fetus and look what is the status. So if you look in the microscopically, the control fetus versus the fetus that came from infected mothers, microscopically, they look exactly the same. So in terms of size and so on. However, if you take now those fetus and we do histology in terms of looking at the development of the organs, this is when the surprise came. When we look at fetus of the mother that's infected, although we do not detect viruses in the fetus, we see a lot of abnormal development in the organs, including the lungs, the vascularity, a lot of bleeding in the tissues. You can see this picture. There is a lot of bleeding in the pericardium, a lot of bleeding in the lungs, and so on. And then now we even are finding with another type of viruses, data that I don't have time to show you, that there is major changes on the organs associated with the immune system, especially the thymus and the liver, abnormalities in the development of those organs due to those infections. But for us in the time, the most surprising aspect that although there was no viral infection, again, I repeat, in the fetus, when we look at the brain of those fetus, there was an abnormal development of the cortex. So while the cortex in the control is thick and very little space in the meninges, the cortex of the mice, of the pups who came from mothers that were infected, the cortex was not properly developed, and there is accumulation of liquid in the meninges. So a maternal infection was having a major effect in the development of the brain, although the virus did not reach the fetus. And that condition is what is very similar to Zika, although in Zika it's thought that also the virus is able to reach the fetus and damage some of the neurons in the development. In our case with MHV68, the developmental effect was by the factors that were produced in the maternal side, especially in the placentas and semina. So we look at the brain of those the pups that came from infected mothers. And is this column that you see here? This is the control in the left, and the right is the pups from infected mothers. And we see a massive inflammatory process going on without the brain of those pups. Yeah. So there is an inflammation that is happening in the fetus, even though there was no viruses. And clearly this inflammation is coming from signals originated in the placenta, as I will show you. So the conclusion of this part is that viral infections of the placenta have detrimental effects on the fetal development. As I mentioned to you, and I hope to show you next time, now we have a lot of data showing that this inflammatory process is not just limited to the brain, it's also happening in multiple organs in the fetus, especially the organs associated with the immune system. So having observed those changes, again, the two aspects, that the placenta is infected, but there is a controlled infection, but also that there is an effect on the fetus, we wanted to understand how the placenta controls the virus and whether those signals are capable of reaching the fetus. So the question was how placenta controls viral infections. And of course, that leads us to a very important pathway on controlling viral infections that even today for COVID-19 has shown to be extremely important, and that is the type 1 interferons. So we know that type 1 interferons have a critical role on interfering viral replication, on controlling the viral replication. That comes the name of interferon, that is to interfere in infection. There are two types of type 1 interferon, interferon alpha and interferon beta. They are extremely important on activating the immune system to stop viral replication, but also they are very important in regulating an infection, in regulating the magnitude of the infection. So, and this is something that I want to keep you in mind for what we were telling at the beginning. What is the differences between an inflammation that kills and inflammation that doesn't kill? And it seems to come from the role of type 1 interferons, because in one side, they are inducing the response against the virus, but in second side, the second response, if you want to call it, is to stop that inflammation, to prevent the chronic inflammation, and that is done through the regulatory effects. So how those regulatory effects, how the secondary response is stopping a massive inflammation is through the expression of anti-inflammatory signals, such as IL-10, PD-L1, and so on. Yeah. So extremely important in the regulation of an excessive inflammation. In case of COVID-19, I've used not very well, what is killing the patients is this cytokine storm and the excessive inflammation that happens in some of the organs. And clearly that seems to be due for the capacity of the coronaviruses to suppress the interferon pathway. So keep in mind about that in view of what we want to show you related to what can happen in pregnancy. So again, to summarize what I just told you, the interferons have these two balancing conditions. One is to strain the immune system in order to stop the viruses from replicating and destroying the tissues, but at the same time to modulate the immune responses so it doesn't become a chronic situation or a cytokine storm that we see today in COVID-19 that may be more dangerous for the patient than the virus itself. How interferon works? Interferon works through the type 1 interferon receptor, activates a intercellular cascade mediated by the STAT2, STAT1. The formation of a complex is the IRF9, And this IRF9 or gas will bind to a specific promoters in the DNA, specific sequences in the promoter of several genes, and activate the expression of those genes. What are those genes? Those genes belong to a big family that is defined as interferon-stimulated genes. Those interferon-stimulated genes are the critical components that mediate all the functions that I just described to you. Activate genes associated with the antiviral response and activate genes associated with the immune regulation. So we wanted to know whether the placenta has the capacity to recognize the virus and respond to the virus. So we look at the placenta, whether there is the production of interferon-stimulated genes by treating trophoblast cells with interferon beta. And we see that indeed this is the case. If we treat trophoblast cells with interferon beta, increasing concentrations of interferon beta, we see a nice dose response in the expression of CCL10. That is an activator of immune cells. We see a very nice response also, dose response on the production of ISG20. ISG20 is this exonuclease, very important in controlling RNA and DNA viruses. And we see a nice dose response in the expression of PD-L1, the regulatory gene that will stop the response. So again, in the placenta, we see the expression of the main interferon-stimulated genes that cover the three aspects of immune responses. Has ISGs that control antimicrobial, have ISGs that control viral infection, and has ISGs that control the immune activation, immune modulators. So having seen that the placenta has the capacity to produce interferons, so the next question was, is this response, are the type 1 interferons responsible for the protection of the placenta against viral infections? So again, this is the work with this incredible team who was working in the viral infections, who I'm going to show you the very elegant work that they have done. So the first question that we have was, what is the role of the placenta as a regulator of viral transmission? Can the placenta stop viral infection and prevent that the virus will reach the fetus? So again, I show you data with MHV68. As I showed you before, the placenta gets infected. You see, this is viral titers. So there is a good, a control amount of viral titers in the placenta of the mouse. I showed you before that was with the cells, the trophoblast cells, human trophoblast cells. So now I'm showing you with the placenta tissue from the mouse. So again, infected, the fetus is not infected at all. Yeah. So again, data that I showed you before, the placenta is able to control a viral infection and prevents the virus to migrate into the fetus. Now, if we do the same experiment, but this time we use a mouse who is lacking the interferon receptor, the type 1 interferon receptor. In other words, it can produce interferon, but it cannot respond to interferon. So it cannot produce interferon-stimulated genes. It cannot activate the expression of the antiviral factors. So when we use this interferon receptor knockout, we infect it with the same amount of virus as the control. We look at what happened into the viral titers in the placenta, and this is what I find. Again, the placenta can recognize the virus. I have not shown you that data because of time. It's mainly mediated by IL-3, IL-4, and IL-8. These receptors will induce interferon beta, but there is no interferon receptor, so there is no ISGs. Okay, so let's see what happened. Contrary to the control, who is able to stop the viral replication if there is no interferon receptor. You see that the virus continues replicating without control. This is a logarithmic scale, so we're going to less than 10 to the 4 to almost 10 to the 10. So massive amount of viral replication in the lack of interferon receptor, type 1 interferon receptor. But here is the major surprise. So in the wild type, as I showed you before, there is no viruses in the fetus, but in the interferon receptor knockout, now the virus has reached the fetus. So definitely, the lack of interferon receptor signals in the placenta is removing the protection for the fetus. So in order to confirm that it's the placenta who is stopping the virus and that the interferon in the placenta is responsible for the control of the virus, we use a combination, a matching combination, where we have a father who is an interferon receptor knockout and the mother who is a heterozygote, but it will function as a normal. It produces interferon and can respond to interferon. If we cross them, what we will have is in the same mother, a fetus who is wild type, so like the mother, it has interferon receptor, but you will have also fetus who are like the father, who are interferon receptor knockout. So again, you have a mother who is able to respond, half of the fetus we will able to respond, and half of the fetus will not able to respond within the same mother. This allows us to dissect what is the maternal component versus what is the fetal placental component. And here we go. If you have fetuses who have the interferon receptor in the placenta and the fetus, with a mother who is wild type, the fetus, the placenta controls the viral infection. However, even though the mother is wild type and has the receptor, but if the placenta doesn't have the receptor, now the placenta cannot control the viral infection, and it's a major increase on viral replication. And here comes about the protection. Again, the mother is wild type. The mother can respond to the virus. It has interferon production and it has the receptor. But if the placenta in the same mother doesn't have the interferon receptor, the fetus is infected. While in the same uterus with the same mother, those who have the interferon receptor, they are not infected. So is the placenta, the conclusion of this is the placenta the one who has the capacity to control viral infections at the implantation site and prevent the transfer of the virus to the fetus. So in other words, to put it in the whole model, when the virus reaches the placenta, is recognized by toll-like receptors, induce interferon beta, binds to the interferon receptor, induce the expression of ISGs, and these ISGs are able to control the infection. If that is not present, then the protection will be gone. So now we know why the placenta and how the placenta is controlling. So the next question is, in reality, what we are confronting all the time. Why some women are more susceptible, why another woman are susceptible, why we put in general this concept that women are in higher risk of mortality during viral infections. So we wanted to understand, again, what will be the trigger in the cases where women are dying during viral infections. So we, again, taking the MHV68, we look at the effect on maternal survival. We look at different doses of the virus, and they always survive. So again, confirming that during pregnancy, the maternal immune system is very strong. Yeah? and it has a high capacity to control the majority of the viruses. And again, we tried with multiple viruses, with multiple doses, the virus always, the mother is always able to control the viral infections and survive. So again, still we wanted to know when will be the cases where that is not happening, when the mother when they will die. And we're going back again to the interferon receptor knockouts. We inject the same virus in the lower concentration, and for our surprise, they died. So this is the pregnant. They died within 4 or 5 days, which is different from the not pregnant. So the same interferon knockout mice, interferon receptor knockout mice, when they are not pregnant and you give them the same virus, there is some mortality, but more than 50% of the mice will survive at the end. That's not the case when they are pregnant. 100% of the pregnant mice, interferon receptor knockout, they will die within 4 days of the pregnancy, within 4 days of the infection if they are pregnant. So what is making the difference between the non-pregnant and the pregnant, of course, is the placental fetal unit that is present. So what we saw is that the loss of target interferon signaling increased maternal mortality during pregnancy. And the major difference between the two groups is the presence of the placental fetal unit. Okay, this is what is making the difference. So we asked the question whether in the lack of the interferon receptor in the placenta, we may have what is called the cytokine storm, or the lack of immune regulation that will lead to a massive inflammation. Yeah. So the first experiment we did is embryo transfer. So we took a mother that is an interferon receptor knockout, and we transferred embryos who will be wild type. In other words, the placenta will be able to recognize the virus and also to respond to interferon. While we did exactly the same, an interferon receptor knockout mother, and we transferred interferon receptor knockout placentas. And this is what we found. So let's go start first what I showed before. If we have an interferon receptor knockout mother, and we transfer an interferon receptor knockout embryos, so they cannot respond to type 1 interferon, we expose them to the virus, and they will die in the first 6 days. Again, there is a difference because of the embryo transfer, but they will die immediately after, a few days after, 6 days after the infection. The infection is happening in day 0, and very kind. So a clear mortality in this. But here comes the surprise. If we inject to the same mother that is interferon knockout, we transfer, that is carrying wild type embryos. In other words, the placenta is able to recognize the virus and respond with interferon to that viral infection. Now the mother survived. So what it means is, and again, if we look at the organs of the mother wild type in the spleen of the mother is able to control the virus. If it has interferon receptor knockout embryos, the amount of virus that accumulate in the spleen is significantly higher. But if she has embryos who are wild type, those viruses are significantly decreased. So what we are seeing here is that in the lack of an interferon response in the placenta, there is a lack of regulation of viral titers. So the placenta becomes a reservoir for viruses, who then start shedding in high numbers into the mother. But even worse that we are finding is that the placenta initiates a cytokine storm, which is then is responsible for the maternal mortality. Yeah. And again, that is the case that you see even today with COVID-19 in cases of mortality where there is the cytokine storm due to the lack of type 1 interferon responses. That is what is happening in the pregnancy. So an infection that is capable to suppress the interferon response or manipulate the interferon response will lead to an uncontrolled antiviral response or excessive or chronic viral replication. And that is, or those are the cases where the life of the mother is in high risk. So a responsive interferon beta pathway in the placenta is essential for the protection of the fetus and the mother against viral infections. For two aspects. One is because it protects, controls the virus, but a second, because it modulates the immune response and will suppress the excessive interferon response. So we see how we are doing in time. Yeah, we're doing time. So one of the aspects that we want to understand is what maintains the signals of interferon, because you don't want to have also interferon beta or type 1 interferons all the time expressed. A chronic type 1 interferon may also lead to complications. So you need to have a basal stimulation to keep the system active, aware that when it comes to infection, you respond quickly and then you suppress quickly. That is the normal homeostasis. So the work that I'm going to show you now is understanding how this type 1 interferon is regulated within the placenta. So one of our interesting findings was that a major regulator of the type 1 interferon pathways is bacterial signals. And that leads us to this interesting and growing concept that is the role of the microbiota. What is doing the microbiota in the majority of the tissues, including reproductive organs? So commensal microbiota controls the development of lymphoid tissues. It controls immune cell homeostasis. It controls the resistance to invasive pathogens and calibrate type 1 interferon function by providing tonic signals. So, and there is plenty of evidence that by disrupting the commensal bacteria environment, you will damage this balance. Antibiotic treatment impairs antiviral responses. There is an increase in the sensitivity to HSV2 of patients who have received a lot of antibiotic treatments. There is an increase of flavivirus infections also when there is antibiotic treatments. And of course, there is a lot of studies showing that preventing antibiotics for influenza instead of really helping, in many cases can make the patient more susceptible to influenza. So disrupting the commensal bacteria may have a detrimental effect on how our body controls the antiviral infection. I'm getting messages and I'm checking just to make sure that you guys have seen the full screen and you can hear me. If something goes wrong in the signal, please let me know. So let's go back to reproduction. So we know that the female reproductive tract do have a commensal bacteria. It's not within the placenta because bacteria are never within the tissues. Bacteria is always in the surface of the tissues. It's always associated with the mucus that is covering the tissues. Yeah. We know that for a study that we did many years ago when we took bacteria that was labeled with E. coli, we put it in the vagina of the mouse, and then we monitor where the bacteria will go. And the green is showing you where the bacteria is localized. So if we deliver bacteria in the vagina, of course, the green fluorescence of the E. coli is in the vagina. But if you look 24 hours or 48 hours, the bacteria has moved the whole reproductive tract of the mice and even reached the ovaries. After 72 hours, the signal decreased significantly. However, it will be present up to 2 weeks, looking by PCR the presence of green fluorescent protein. What do we learn from this image? So let's get all the message that we see from this experiment. First of all, as I mentioned, bacteria from the vagina has the capacity to move from the reproductive tract without any problem. The second aspect that we see at the 72 hours is that the mucosal immunity of the uterus is extremely efficient, has the capacity to control that E. coli. It disappears in 72 hours, the excess of bacteria. That's the reason we don't see the signal. So powerful mucosal immunity in the uterus. So although the uterus, and not only control, it will eliminate that bacteria. But the second aspect that we learned from this experiment, the PCR, that we still see detectable bacteria after even 2 weeks, is the fact that the bacteria can remain present in the uterus and will remain for months or forever. And again, now we know that the uterus has a normal mucosa. The uterus is not a sterile organ. So if we put it now in the picture, this is the reality that also happens during the pregnancy. The placenta itself, as I mentioned to you, is a tissue that shouldn't have bacteria, and it doesn't have bacteria. Because if bacteria reach the placenta, you have an infection, like any tissue. The gastrointestinal tract, who has more bacteria than other tissue, you will never see bacteria within the tissue, never. The bacteria is only in the surface of the gastrointestinal tract. And this is what happens also in the uterus. The bacteria is in the surface of the uterus associated with the mucin. However, the bacteria is producing factors, and those factors are the placenta and potentially reaching the fetus. So the question was, if the bacteria who is in the lumen of the uterus is secreting factors, how those are recognized by... I think we have a microphone that is active with somebody.
Your sound, Huawei Mate 30. Yeah, please.
Sorry.
Are we okay?
Yeah, okay.
Okay, thank you.
Come.
Okay, good. So we have 10 minutes. So what I'm showing you here is that the signals from the bacteria coming from the uterus, as well as the signals from bacteria coming from the maternal circulation. There is, for example, LPS in the blood of the woman, a pregnant woman. It's interesting, the LPS also increases during pregnancy in the circulation of pregnancy. All those signals coming from bacteria in the body of the mother. The question was, are recognized by the placenta? And if they are recognized, what are they doing? So we wanted to know what is the role of bacterial-derived signals for the protection against bacterial infection. So it doesn't matter where is the bacteria is releasing factors that we think will be also protected by infections because of the data that I showed you before. If there is no bacteria, if you break bacteria, there is a risk of having viral infection. So this is what we did. And again, disrupting. Let me pass this so we go. I mentioned to you yesterday that the placenta has all the toll-like receptors, and therefore can recognize bacterial products. So we use LPS, a well-known bacterial product from different types of bacteria, and we expose in normal concentrations to trophoblast cells, and we see what type of cytokines or type of response. The interesting aspect is that when we treat trophoblast cells with LPS in normal concentrations, we don't see the induction of the classical pro-inflammatory cytokines. There is no increase in TNF-alpha, and there is no increase in interleukin-1 beta. What we do see is that the LPS in those normal concentrations, or the physiological concentrations, is the expression. Here we have somebody talking without muting the microphone. So what we see is the induction of
华为 Mate 30的是谁？赶紧出去吧，出会吧，严重干华为 Mate。
And I cannot mute everybody. I don't know if Alisa can
Can kick.
Today things are not working so well. We are learning all the problems that the system can do. The technology is fascinating, but it still has its limitations. So let's go back here. So what we found fascinating is that the signals from bacteria really are maintaining the expression of interferon beta. And you can see here a dose response. Trophoblast cells treated with LPS, 10 nanograms, you start increasing the expression of interferon beta messenger RNA. You do see the expression at the protein level of interferon beta enhanced with LPS 72 hours. But more interesting, not only we see an increase in the intracellular protein interferon beta expression, we see also an increase in the secretion of interferon beta by trophoblast cells treated with LPS. So if we put it in a graphic, LPS coming from bacteria binds to TLR4, induce interferon beta, and maintain that expression of interferon beta. And it's specific to interferon beta, in the placenta at least. We don't see interferon alpha. Nice dose response to interferon beta. And more important, I want you to look at this figure. Yeah, and again, we know now how is the mechanism of regulation. Because of time, I'm not showing you. The interferon beta goes up in the messenger RNA in the first 120 minutes, and then goes down. Yeah, this control is essential to maintaining the balance. And we will continue more time, it follows the same cyclicity. It goes up, it goes down, it goes up, it goes down. Yeah. It's the same in the mouse. If we treat the mouse, the wild-type mouse with LPS, take those placenta, we see a lot of interferon beta produced. Yeah? And the pathway that it follows, again, I'll go quickly through this because already I have shown and it's published. The pathway that follows is LPS bind to TLR4, activates TBK1, IRF3. IRF3 is responsible of the induction of interferon, and then it will bind to its receptor. This basal production of interferon depends on TLR4 and TRIP, because if we have a TLR4 knockout or a TRIP knockout, the production of interferon beta is completely suppressed. So what we know now is a baseline interferon beta expression in the placenta is maintained by LPS, and this is extremely important for the modulation of the maternal immune system. And we think that this interferon beta expression, this basal maintained by LPS, is again extremely important in the first response against a viral infection. So the microbiota induce the interferon beta. If a virus comes, the interferon beta is already ready. If the virus is recognized immediately, interferon beta will bind to receptor and will induce the expression of ISGs, and these ISGs will stop the virus, but also will prevent the cytokine storm, which is extremely dangerous. When this becomes affected, so, and this is the interesting thing, is the communication between the two systems. So what we show is in this experiment quickly, because of time, any type of infection that will disrupt this equilibrium will lead to the termination of the pregnancy. For example, if the mice is infected with the virus, in this case, MHV68 or herpes virus, if it's infected with the virus, and then 7 days later, we treat the mouse with a low concentration of LPS, and we look what happened, this is what we found. Again, let me show you. The virus, as I showed before, by itself doesn't do anything to the pregnancy in terms of termination. LPS and low concentration has little effect. However, this double combination, the virus infecting the placenta, and then the same signals of the commensal bacteria, the same signals of LPS, which before did not do anything, now you will have a total termination of the pregnancy. And what we see the difference? The combination of a viral infection and the LPS is inducing what we call the cytokine storm, an uncontrolled inflammatory response that terminates the pregnancy and potentially kills the mother. And again, this is showing all the characteristics. If the virus suppresses the interferon pathway, which is, this is the case in this virus, many viruses like corona, the way they are able to escape immune surveillance is by suppressing the type 1 interferon. So MHV68 was able to suppress the type 1 interferon in the placenta. And when we give the LPS now induced a massive cytokine storm that terminated the pregnancy. So the message is the following. Viral induced suppression of interferon beta pathway leads to fetal demise. So, and the fetal demise is characterized by a pro-inflammatory cytokine response, or what we call today the cytokine storm that will induce fetal demise. So the conclusion, the final conclusion of this is what we proposed several years ago, that many of the pregnancy complications, such as preterm birth, are a result of the double hit or the two-hit hypothesis, where the commensal bacteria is maintaining the balance, the regulatory balance, the immunological balance at the implantation site by stimulating interferon beta, and therefore maintaining a tolerance and maintaining a protection, but also preventing an uncontrolled inflammatory response. If something breaks that balance, such as the viral infection that suppress the interferon response, now you will have a break in what we call the interferon-mediated tolerance and a massive inflammatory response that will terminate the pregnancy. When that massive inflammation reach also the mother, it puts the mother in danger and can also be the cause of mortality. So I finish now with the overview. The placenta and the pregnant woman are not immune suppressed. The placenta plays a central role during pregnancy by responding to viruses for two aspects. One is by controlling viral replication, preventing the virus will reach to the fetus, and also preventing that excessive virus will reach the mother. In order to accomplish that, the normal microbiome, the microbiota, is maintaining a basal tone or baseline expression of interferon, that one interferon that will bind to the interferon receptor, will induce ISGs. And you have the ISGs that control viral replication, that control viral infections, but also have an important role on maintaining an immune modulation, on preventing massive cytokine storms. If, and again, protects the fetus and educates in a proper way the development of the fetal immune system. If we remove the bacterium, we disturb that basal communication, viruses will reach, the interferon response will be affected, viruses will reach the placenta, will multiply without control, they have the capacity to further suppress ISGs. If they suppress ISGs, they suppress the control of the virus replication. As the virus continues replicating, will also alter the immune regulation, and then you will have a cytokine storm that will terminate the pregnancy, but also will have an effect on the mother. If the pregnancy survives, for example, we are now knowing, we know now, we have a lot of data now showing what is happening to the development of the fetal immune system, who will be future talks. So the placenta is a preferred site to viral replications. When a viral infection breaks the immune regulatory function of the placenta, leads to enhanced inflammatory response. We have now corona is exactly the best example for what we have been talking in the placenta in the past. Yeah? And that will lead to detrimental consequences for the mother and the fetus. Neonatal complications, this is the new area that we're focusing. Neonatal complications such as autism, microcephaly, allergies, abnormal responses of the newborns to vaccination is a consequence of altered or inflammatory responses at the maternal fetal site during the pregnancy. And with that, I will stop here, and I will be happy to answer any question. If we still have time for questions. Let me see if I can put this in big. I have to stop sharing. If I can stop sharing. Okay. I will be happy to answer any question. If you don't mind, Professor Young, to read me the questions, because I don't see the chat for this moment.
Okay, I can help you. Okay.
Thank you.
So the first question is raised by Hu Hai. So maybe you can raise your question by yourself, Hu Hai. Hu Hao. Yeah. Sorry.
Hello, Professor. Can you hear me?
Yes. Oh, I know the chat. Okay. Yes.
Hello, Professor. It's really a wonderful lecture. And could you please introduce the advanced research development about the pathological effect that COVID-19 imposed on pregnant women and fetus? Thank you.
Yeah. So indeed, as I mentioned at the beginning, we still are learning what is going on with COVID-19 pregnant women. What I can tell you, everything, the mortality or the complications of those patients is associated with the symptomatology. So if the patient is showing, a pregnant woman infected with SARS-CoV-2 is showing some symptoms of inflammation, she is in high risk. there are multiple patients who were infected with COVID-19. There was no signs of inflammation. The virus was properly controlled. They have no problem, they would survive. At the beginning, people were talking that there is no infection of the placenta, that the virus is not able to enter the placenta. But I think that still is a subject of debate. This virus mutates a lot. It has different ways how it could infect the placenta. In the majority of the cases, I'm convinced the placenta will prevent that the virus will reach, will block the infection. But it seems there are some cases where the placenta gets infected. And I think that when the virus reaches the placenta, that is the case where the mother is in risk, because the virus has a high capacity to suppress type 1 interferon responses. It does in the lungs. We have a paper just coming out in the Journal of Leukocyte Biology, where we describe the pathways, how the virus seems to block the type 1 interferon. Once it comes out, I will be happy to share with you. So to summarize what I'm trying to tell you, there is a lot of questions about pregnancy, but our data suggests that in the cases when the virus may reach the placenta, you will see a symptomatology, and if you see symptomatology, then those pregnant women are in risk. If the virus doesn't reach the placenta, and if it's well controlled, a pregnant woman has no risk with COVID-19.
Dr. Gil, can I add a little bit more?
Please, please.
Because just recent study shows that there are some viral particles of COVID-19 cases in the placenta. There were quite a bit of confusion, but I think most recent studies are showing the presence of virus. And clinically, although mother does not have that much COVID-19 symptom, this patient with the COVID-19 positive cases, placenta looks awful. So I think your theory really hits well. So placenta damage is ongoing even before the clinical manifestation of the mother. So I saw that pathology study and was very excited about it.
Wow, thank you, Joanne. That is, I would love to see again. I think I saw a couple. There is a lot of things coming out in terms of the pathological. I had this discussion, a lot of discussion about the infection, and I think we talked also a little. As you know, the study from Roberto Romero that they claim that there is the receptor is no enter. We did with different models that the virus can enter, but again, I'd love to hear from you the data. There is cases of placenta. I'm convinced that there is cases of placenta that get infected, and it makes a big damage if it does infect. Thank you, Joanne.
You're welcome.
Next question is raised
You
by
just talk about the reaction of the mother's immune system after infection, that the fetus immune plays a role in fighting the virus. Very, very good. Yes, yes, absolutely. You know, the fetus is not in standby, just looking what is happening. No, from the beginning is activated. We have done studies with Zika, again, infecting Zika later in the pregnancy, so we will not have the direct effect on microcephaly. And it's fascinating because we look at the immune organs of those fetus and they are activated. Yeah. So when there is an infection, the fetus is able to respond in its own way. Yeah. Whether it's protective, not protective, we don't know, but the fetal immune system is changed, is modified. What we are trying to understand is those modifications, those, let's call it those responses that are happening in the fetus during that, during the pregnancy, whether it translates into abnormal or modified immune system in the neonatal. And some data suggest that that's the case. Again, this is preliminary data. We have taken pups born from infected mothers with Zika. We challenge them when they are adults again to Zika, and surprisingly, the response is completely different from pups coming from moms who never were infected. So the fetal immune system is able to respond to the infection that is happening in the mother, and that response seems to be priming or reprogramming the immune system that will alter the response later. In some cases, maybe a better response. In some cases, maybe not. That is still we don't know. What we know is that the responses are different. Professor, what treatment measures do we have to improve maternal fetal immunity and antiviral ability? Outstanding question, and that is a question for all of us who work in the field. At the present time, what I can tell you without any doubt, that is vaccination is the best protection. For many years and in many places, still there is the dogma that you should not vaccinate pregnant women because it's dangerous for the fetus and the mother. It's absolutely not true. The concept of not vaccinating pregnant women was in the idea that the pregnant woman is immune suppressed. And now we know very well that that is not the case. So you should vaccinate pregnant women. That is the best protection. And in addition, when the pregnant woman is vaccinated and develop immunoglobulins, those immunoglobulins are going to be transferred to the fetus in the second trimester. And those immunoglobulins going to the fetus are going to provide the protection as well as the training for the fetal immune system. So at the present time, the best protection that we have is vaccination. Antiviral treatments, that's a little more complicated. Yeah? And again, it's controversial in terms of, because many of the antivirals that exist, they use normal mechanisms of the cell for its function, like proteasome inhibitors or RNAs and so on. So they can inhibit the viral replication, but also they can damage the function of many cells. And we know that the fetal development is very sensitive. We cannot discuss that. Yeah, it is true. So damaging some normal functions, it could be detrimental. So antivirals is a challenge. Although there are patients with HIV who have received antivirals and without apparently any effect, but that is a delicate subject still. Yeah, vaccination, definitely no. So that is the best tool that we have now. And again, Joanne, if you want to add something, please feel free. Okay.
Oh, I'm sorry, I was on mute.
It's okay.
Yeah, it's vaccination is, I think, probably the best way to do. So usually when the pregnant woman gets into that question, then physicians are nowadays recommending to have a vaccination during the early first trimester to get the best benefit during the second, third trimester, or basically at any time if the patients are missing it. I think that's probably the best strategy in this point. But then some using the antiviral drugs, those can be still need to go through a lot of checks and balance things. And so those are still need to be studied. But then I think that probably what we have right now, the vaccination is probably the best way to go.
Perfect, thank you. How to treat a pregnant woman if she's infected with influenza virus? Yeah, so in general, again, we say what the emphasis, what Joanne mentioned, vaccination. Yeah, we enter the season of influenza. Pregnant women or women who are planning to get pregnant, vaccination is the best thing. In general, a woman getting influenza is not that she's going to be in a high risk of anything. I mean, I think the majority of women get a cold, yeah? So that is not a problem. The problem is when we have the situations of pandemic where the viruses are extremely aggressive, and they are, especially influenza virus is an RNA virus. So all RNA viruses, evolutionary, as you know well, all the RNA viruses are the faster modifying viruses. DNA viruses mutate less. Why? Because DNA, as you know very well, the DNA has checkpoints. So when the DNA is replicating, if there is a mutation, it will try to fix. So it will fix the mutations. DNA viruses mutate very little. RNA viruses, on the other hand, there is no checkpoints for replication. So every time when a cell dies and releases all those viruses, is releasing a high percentage of viruses who already have acquired mutations. Because RNA replicate without control in terms of the mutations. They acquire very quickly. That's a problem with influenza. So that is the reason every season we have different ones. As long as we maintain with vaccination, control the viral replication, influenza during pregnancy or the cold during pregnancy, it shouldn't be a risk, and it's not a risk. The maternal immune system will control. What we want to prevent is that the influenza will get chronic, because then the risk of the influenza reaching the fetus, reaching the placenta, increase. Yeah. Again, the best source that we have today is vaccination. Can you talk about microbial host molecular exchange and its effect on early life? Yeah, if I understand correctly, the question is, what is the effect of the microbiome in early life? This is a fascinating area. There is a lot of ongoing studies. We are learning in many aspects what is going on. I just summarize very quickly because, again, we don't have much time, and it's an area that we are just learning. What we know without any doubt is that bacterial signals do reach the fetus. Not the bacteria itself, I repeat, bacterial signals do reach the fetus. And that bacterial signals are essential for inducing the maturation, the development of the fetal immune system in the normal way. Because when we come out from the womb of our mothers, when the mother delivers, the baby comes out to a world that is surrounded by bacterial microorganisms. Remember, 200 years ago, 100 years ago, or today in many places in the world, deliveries don't happen in sterile operation rooms. Woman delivers in the field. Woman delivers the babies in the homes where there is plenty of bacteria. So if the immune system of the fetus will not be already prepared to deal with that bacteria, we will not be here as a species. We will be gone. The fetus will die. It's true. After birth, the mother will continue transmitting immunological signals through the milk that will confer more protection. But that first shock of coming out into the world and an environment with bacteria, the training that the commensal bacteria was given to the fetal immune system is the critical step for the survival. So that's what we know about the signals from commensal helping the development of the immune system. The mechanism of action, what is the leading to those things, is what we are learning now. Okay, so I think with that we have completed questions. Professor Liao, I don't know if you want to have a picture of everybody with the cameras.
Yes, we can. Yeah. So everybody, yeah. So to light or today is really the last session of the international course. Okay, so maybe during these times, Professor Gil and Professor Clark give us totally 8 wonderful lectures, which really let us learn, systemically learn the knowledge both the basic and clinical reproductive immunology. I believe all of us really learn a lot, and we are really enjoying their wonderful lectures. So although this is a trial, but it proved very successfully. So I hope we will continue this kind of international course. Next year, maybe we can offline. Yeah, so finally, I would like to express all of us sincerely thanks to Professor Gil and Professor Clark for your hard work and wonderful lectures for all of us. Thank you. So maybe before the ending of the lecture, maybe I invite to Professor maybe give us a very short remarks. Okay, is it okay? Professor Gil, give us.
I always like to say ladies first. Professor Joaquin.
Dr. Mor is always wonderful.
If you allow me to be a gentleman, I still think the gentlemen are politically correct.
Yeah. First of all, I was very happy to involve with this lecture series and meeting with the students for multiple times. I continue the whole series of reproductive immunology lectures. So it was kind of not only giving the lecture to the student, kind of fun, but then it was very rewarding to myself as well. And then Dr. Mor's lecture was also very helpful to everybody, and it kind of reviewed the whole process of basic reproductive immunology. So thank you for Dr. Mor's lecture as well. And thank you for listening to this lecture. Hopefully we can continue this series next year as well. Thank you, Dr. Liao.
Thank you.
Thank you so much. And again, it has been a pleasure of participating in this program, especially with my good colleague, friend of many years, really a great friend, Dr. Joan Joaquin, which I learn from her every day, and it stimulates the research. I want to thank Professor Liao to put this together. I mean, I tell you the truth, when you told me that you want to put this Zoom course, I was not so excited because, as you know, I like to talk to people. I don't like to talk to empty chairs, but it has been a great experience. The participation of all the students, the faculty, and so on, the questions has just excited me every time coming. And although I am talking to a camera who is looking at me all the time, I still feel that I'm talking to some human beings almost 4,000 miles from here. Mm-hmm. I love doing this, but I love better to do it personally. The warm relationship with you, with everybody of you is a critical component of what we do. But again, this has been an incredible experience. I want to thank all of you because I know it's late in the evening when you're participating. So congratulations for staying so late. Thank you for your questions. Thank you for your participation. And I look forward again to do it again in the future. Professor Liao, Professor Joaquin, thank you so much for making this course an outstanding experience.
You're welcome. Thanks.
Welcome. Thank you. So last work is take a picture together. Okay. Yuan Yao, Wang Huan, are you ready?
Yes. Yes.
Please. Also say 1, 2, 3. What? Okay.
Okay. 1, 2, 3. Cheers. Cheers.
Yes.
Uh,
If you finish, you say you told us. Okay.
okay.
Finish.
Yes, yes. I
Okay. Last.
mm,
Okay. So
okay.
that's the ending of this international course. So we have to say goodbye to everyone. Have a nice day. Yeah.
Goodbye,
So
and
Bye.
thank you
So
so
be-
much for coming.
okay.
Good to see all
Thank
of you. I hope
Yeah.
to meet you in the next future. Thank you.
Yeah.
Thank you.
Bye-bye.
Bye.
Bye-bye.
Bye.